I'd like to welcome everybody to this week's edition of the Extracellular Vesicle Club. And this is indeed a very special edition for us because it is the start of a collaboration with SOCRATES, the society in Singapore, that is actually one of the oldest societies for EV research in the world. And so, as some of you know, this journal club started as something that I held at Johns Hopkins for about 4 years before the COVID-19 pandemic started. And when COVID-19 began, we thought, why don't we just turn this into a worldwide kind of journal club, also research club. We don't just have to present journals. And so we began this in March of 2020. And a few months ago, the International Society for Extracellular Vesicles came to me and said, why don't we maybe make this into an ISEV event, not just something that your lab is holding anymore. So I'm Kenneth Witwer. I'm the chair of science and meetings for the International Society for EVs. And I, as I said, I've been helping with this club for about a year now, since we went virtual. And unfortunately, because it began as a local institutional thing, we've usually had it at a time that is not very convenient for those of you who are in the Asia Pacific region. But with this new collaboration, we're excited to be having some events that are going to be much easier for some of you to join. It won't be in the middle of the night for you. So Minh, thank you so much for helping to organize this session. And I'd like to hand this over to you to introduce SOCRATES and then to introduce our very special presenter today.
Thank you so much, Ken. So I'm Minh. I'm from National University of Singapore. I'm the assistant professor here and also the vice secretary of SOCRATES. So you have, some of you have known SOCRATES, but maybe not many of you know us. SOCRATES is a society for clinical research and translation of extracellular vesicles in Singapore. It was registered in 2013 and it's a non-profit organization that aims to promote clinical research, application and translation of extracellular vesicles, to organize events, meetings related to extracellular vesicles, to raise awareness and publicity of extracellular vesicles, and to become a platform for collaboration and combined research, not only in Singapore, but also with international collaborators. So we are very happy to be part of ISEV and to collaborate with Chen in order to organize this EV club. So if you are interested in joining SOCRATES, if you haven't, I send you a link in the chat box. You can feel free to contact us and join us if you're interested. Today, we're very happy to have Shawn as our first speaker. So Professor Shawn Chen is professor of medicine and technology at our school, the Yong Lin School of Medicine. And he's also a professor at Faculty of Engineering at the National University of Singapore. His current research interests mainly focus on theranostics, and he's as part of his research, he worked on membranes, cell membrane vesicles that are very similar to extracellular vesicles. So we hope to hear from him today about his recent research on these vesicles and how he used that for cancer treatment and different types of therapeutics. On to you, Shawn.
All right, thank you, Ken and Minh, for the very kind invitation. I feel a little intimidated because EV research or extracellular membrane, I cannot even pronounce it, and cell membrane vesicles are not my major research efforts, but I feel this is a booming field. It's very exciting, and there is a lot to explore. Otherwise, using conventional nanomaterials like liposomes may or may not accomplish. So, as I already told Minh and Ken, because I was nervous and I was distracted a lot, so I pre-recorded my talk. So I hope it's something we can discuss after this short presentation. So I'm going to share the screen now. I really appreciate the kind invitation from the Society for Clinical Research and the Translation of Extracellular Vesicles, Singapore, and the EV Club of the International Society for Extracellular Vesicles. We work to some extent on cell membrane vesicles instead of EVs. Hope the Society and the Club will consider expansion to include biomimetic cell membrane-coated nanoparticles and vesicles. This photo was taken 2 years ago, before the pandemic. It was the 10th anniversary of my lab at the National Institutes of Health. At that time, we had over 30 people. Last year, during the pandemic, I chose to close my lab in the US and moved myself to Singapore. It's particularly challenging to build a new lab from scratch as of now. Everything is kind of slow, as most of the data presented here were from the work done before. My lab used to work on a number of subjects, such as nano biosensors for ultrasensitive biomarker detection. The biomarkers are mostly proteins. We try to push the detection limit. By increasing the sensitivity, we manage to maintain the specificity and accuracy of detection. As a chemist by training, we also develop various molecular imaging probes for in vivo detection of biomarkers. The imaging modalities include radionuclide imaging, such as positron emission tomography, single photon emission computed tomography, and non-radionuclide-based imaging methods, such as various optical imaging techniques, like near-infrared fluorescence, photoacoustics, Raman, OCT, as well as ultrasound, MR, and multimodality imaging. Here is a quick list of a number of multi-imaging probes we have pushed into clinic for first-in-human studies and for early phase clinical trials. We also worked on various therapeutics, such as radioligand therapy targeting somatostatin receptor, prostate-specific membrane antigen, PSMA, fibroblast activating protein, FAP, and so on. Some relatively simple nanoformulas are also in the process of IND-enabling studies, but they are more costly and challenging in translation. We are also developing both inorganic and polymeric nanomaterials for theranostic applications. They may have some unique properties that small molecules and biologics do not have. After self-promotion, let's come back to today's topic. First, let's compare liposomes and the EVs, and then discuss why we want to study cell membrane-coated particles and cell membrane vesicles. On the left is a scheme of pegylated liposome that's split in quadrants. Lipophilic drugs can be loaded in the bilayer membrane. Ligands can be incorporated to increase tissue targeting specificity. Hydrophilic drugs like doxorubicin can be loaded in the lumen of liposomes. Onpattro, the first FDA-approved siRNA-loaded lipid nanoparticles to treat polyneuropathy, is made of ionizable lipids, cholesterol, pegylated lipids, and helper lipids. On the right side, extracellular vesicles as a drug delivery system, proteins, hydrophilic drugs, and the nucleic acids such as miRNA, siRNA, and mRNAs, etc., can be loaded in the lumen of vesicles, whereas targeting ligands, membrane proteins, and the lipophilic drugs can be incorporated in the membrane. EVs contain the full repertoire of cellular lipids and are particularly enriched in lipid raft components like sphingomyelin, cholesterol, and the lysophospholipid. A higher degree of complexity can be achieved with EVs in comparison with mixing individual components in liposomes. Due to the presence of biomolecules in the membrane and core, additional binding pockets may be present in EVs for drug loading. However, EVs also have a number of limitations. For example, loading anything into preformed EVs can be a challenge. Since EVs are part of biological systems, their compositions will be influenced by the cultural conditions of the parental cells. This presents a challenge for manufacture and quality control, as a particular EVs is a snapshot of the cells at the time of production, which may vary from cell to cell, from culture condition to culture condition, and over time. In terms of production and harvesting of EVs, it is also highly challenging to scale up. Thus, we and others seek to develop cell membrane-coated nanoparticles due to their unique abilities of biomimicry and biointerfacing. There are various methods in the functionalization of cell membrane-coated nanoparticles, including lipid insertion, membrane hybridization, metabolic engineering, and genetic modification. The idea of applying cell membrane to coat nanoparticles most likely stemmed from the need to find alternatives to pegylation. PEG, or polyethylene glycol, reduces renal clearance, increases circulation half-life. PEG also improves drug solubility and decreases immunogenicity. However, anti-PEG antibodies can limit the activity of pegylated drugs and may compromise the activity of other effective therapeutic agents. One alternative to pegylation is a top-down biomimetic approach in which the particles are functionalized by coating with natural erythrocyte membrane or RBC membranes, including both membrane lipids and associated membrane proteins for long circulating cargo delivery. This seminal work by Liangfang Zhang from UCSD opens up a new field of cell membrane engineering. Inspired by the way that most enveloped viruses hijack a host cell membrane and subsequently release from the infected cells by a budding process that requires cell membranization, we reasoned that we could genetically engineer viral antigen to harbor into cell membrane, followed by the formation of uniform spherical virus mimetic nanovesicles, or VMVs for short, that resemble natural viruses in size, shape, and specific immunogenicity. As a proof principle, we established both 293T and HeLa cells that stably express an epitope of L2 protein of human papillomavirus 16, HPV16, on the surface of cellular membrane. The construct had a signal peptide sequence from membrane-targeting integrin protein, an epitope sequence from HPV16 L2 protein, and a linker of transmembrane peptide sequence. They were genetically fused to the N-terminal of EGFP, forming SIG16L2 EGFP recombinant protein. Abundant SIG16L2 EGFP signals were found in both nuclear membrane and the plasma membrane, whereas 16L2 EGFP without signal peptide only existed in the cytoplasm. Vaccination of mice with VMV-L2 could not only induce high level of antibody titers to the L2 protein, but also elicit many times higher total antibody titers and about 1 order of magnitude higher neutralizing antibody titers compared with free L2 peptide group in the presence of alum adjuvant through intramuscular injection. Motivated by the goal of developing a method to use VMVs or virus mimetic vesicles as a broad vaccine delivery vehicle for rapid response to emerging pandemics, we tried H1N1 virus. We prepared VMVs to display hemagglutinin HA glycoprotein from influenza A H1N1 virus. In panel C, VMV-HA can efficiently agglutinate red blood cells like active influenza virus. Panel F shows that VMV-HA treated by trypsin induced higher HA titers than ones without TMCK trypsin treatment. VMV-HA resembled live influenza virus in shape, size, and specific function. VMV-HA could trigger high level of serum IgG titers via intramuscular injection with or without alum adjuvant. The total antibody titers induced by VMV-HA in alum adjuvant was comparable to that elicited by standard HA glycoprotein recombinant from baculovirus. Infectious diseases are a leading cause of mortality worldwide, with viruses and bacteria in particular having enormous impacts on global healthcare. One major challenge in combating such diseases is a lack of effective drugs or specific treatments. In addition, drug resistance to currently available therapeutics and adverse effects caused by long-term overuse are both serious public health issues. A promising treatment strategy is to employ cell membrane mimics as decoys to trap and to detain the pathogens. Here, we developed an engineered cell membrane nano decoy for COVID-19. Briefly, the nano decoys were established in 3 steps. Step 1, we genetically engineer ACE2 on 293 T cells. Step 2, we collect cell membrane nanovesicles from engineered 293 T ACE2 cells and the human myeloid mononuclear THP1 cells. In step 3, we fuse the resulting 2 nanovesicles together. In this design, the nano decoys inherit abundant ACE2 and the cytokine receptors from the source cells, enabling effective intervention of COVID-19 by concurrently neutralizing SARS-CoV-2 viruses and the inflammatory cytokines such as IL-6 and the GM-CSF. To extract cell membrane nanovesicles, the intracellular content was removed by a combination of hypotonic lysis, mechanical disruption, and the gradient centrifugation. Subsequently, 293T ACE2 cells and the THP1 cell membrane-derived nanovesicles were prepared. After that, ACE2 vesicles and the THP1 vesicles were mixed, sonicated, and repeatedly extruded through nanopores to form the nano decoys. Through Western blotting, we confirmed that the nano decoys preserved critical receptor proteins responsible for virus and cytokine binding, including ACE2 for SARS-CoV-2, CD130 for IL-6, and CD116 for GM-CSF. We tested various pseudo-typed coronaviruses, such as pseudo-SARS-CoV-2, pseudo-SARS-CoV, and the pseudo-SARS-related coronaviruses. We found that the pseudo-virus infection was significantly inhibited by ACE2 vesicles and the nano decoys, but not by 293T vesicles and the THP1 vesicles. indicating the broad-spectrum antiviral capability of the nanodecoys. Authentic SARS-CoV-2 was also used to test the antiviral ability of nanodecoys. To accomplish this, monkey kidney viral E6 cells were incubated with SARS-CoV-2 in the presence of nanodecoys, and the expression level of viral nucleoprotein or AIM protein was evaluated by immunofluorescence. Similar to the pseudovirus infection results, ACE2 vesicles and nanodecoys showed better antiviral effects than 293T and the THP1 vesicles. Here, in solution containing known amounts of IL-6 and GM-CSF were incubated with nanodecoy nanoparticles. After the removal of nanoparticles, residual cytokines in the supernatants were quantified by ELISA. Nanodecoys are as effective as THP1 vesicles in removing IL-6 and as effective as ACE2 vesicles in removing GM-CSF. To mimic the infection-induced inflammation, lipopolysaccharide, LPS, was used to stimulate THP1 cells to upregulate inflammatory cytokines. After LPS treatment, both IL-6 and GM-CSF levels were significantly higher. Nanodecoys were effective in neutralizing these cytokines in a dose-dependent manner. We fluorescently labeled the nanodecoys and studied the biodistribution of such fluorescently labeled nanodecoys. After inhalation, we see over time the concentration in the lung region is reasonable. We then used an acute lung inflammation ALI mouse model. The mice were first intratracheally inhaled with LPS. At 2 hours post the challenge, the mice received intratracheal inhalation of nanodecoys. Another 8 hours later, lung bronchoalveolar lavage fluid, or BOF, was collected for cytokine measurement. In the BOF, IL-6 and GM-CSF levels were significantly higher after LPS challenge, and the nanodecoys effectively decreased the cytokine levels. The H&E staining at 24 hours post the LPS challenge showed alveolar wall increasation, alveolar cavity disappearance, vascular dilation and congestion, and the alveolar inflammatory cell infiltration. Notably, the nanodecoys significantly suppressed the lung injury, suggesting the potential of nanodecoys in the inhibition of COVID-19 associated immune disorder and lung injury. Now, let's switch gears from infectious disease to cancer. Effectively activating host immunity against cancer has already become a promising therapeutic strategy. However, cancer cells resist to host immune response through both innate and adaptive immunity. Especially CD47 on tumor cells protects them from macrophage phagocytosis through interaction with SIRP-alpha, while PD-L1 interacts with PD-1 to reduce T-cell-mediated tumor killing. Here, we report that genetically programmable fusion cellular vesicles, or FUSE-CVs, displaying high-affinity SIRP-alpha variant and PD-1 can disable both mechanisms. Dual blockade of CD47 and PD-L1 with fusion CVs synergistically promote antigen presentation and activate anti-tumor T-cell immunity. Here, 41 cells were treated with original CVs or SIRP-alpha CVs and then co-cultured with raw 264.7 macrophages. Confocal images showed the phagocytosis ability of macrophages in a dose-dependent manner after the sub-alpha CVs treatment. Panel C and D shows the in vivo effects of sub-alpha CVs on immunomodulation was studied in a 41 tumor model. A significant increase in CD11C positive cells were observed, and these cells showed high-level expression of CD80 and CD86. Panel E and F, a reduction in FOXP3 plus regulatory T or Treg cells and myeloid-derived suppressor cells, MDSCs, within the tumor microenvironment was also observed, suggesting the reversal of tumor immunosuppression. Here in panel G, similarly, PD-1 vesicles blocked PD-1, PD-L1 pathway and activated T-cell immunity, which was indicated by a significant increase of interferon gamma level. After the treatment with PD-1 CVs, significant increases in CD4 positive T cells and the CD8 positive T cells in the tumor, and the interferon gamma positive CD8 positive T cells in draining lymph nodes were observed. The B16F10 tumor mice were injected intravenously with 3 doses of the formula every other day. The sub-alpha CVs and the PD-1 CVs had limited effects on the tumor growth inhibition. In contrast, the fusion CVs significantly prevented tumor growth, and it is better than the cocktail administration of sub-alpha CVs plus PD-1 CVs. Flow cytometry results showed a significant reduction in CD206 positive M2 macrophages and MDSCs, suggesting a downregulation of tumor immunosuppression. Meanwhile, the fusion CVs increased the percentages of CD8 positive T cells within the tumor microenvironment and the interferon gamma positive CD8 positive T cells in the draining lymph nodes by concurrently blocking innate and adaptive immune checkpoints, i.e. CD47 and the PD-L1. The fusion CVs significantly promoted the antigen cross-presentation and boosted anti-tumor T cell immune response. We also tested the fusion vesicle formula in a post-surgical 41 tumor model. After incomplete excision of tumor by surgical removal, the mice were intravenously injected with 3 doses of different formulas every other day. Again, the fusion CVs had more effective inhibition of tumor recurrence. Histology also showed much lower lung metastasis rate, with effective control of tumor recurrence and lung metastasis. Most mice receiving fusion CVs were still alive on day 60 post-tumor inoculation. To develop dendritic cell membrane vesicles, we generated a recombinant adenovirus expressing GFP or OVA model antigen. We also transduced immature DC2.4 cells to express the membrane localization model antigen GFP and or OVA and differentiated those immature cells into mature dendritic cells. The DC nanovesicles were isolated by multi-step density gradient ultracentrifugation and sheared to a desired size using membrane filters. The cryo-EM showed that the nanovesicles had a uniform vesicular morphology. The dynamic light scattering DLS analysis revealed the average diameter of approximately 110 nanometers. Western blot confirmed the major functional membrane proteins were present on DC-NVs like MHC1, ICAM1, B71, B72, and lymphoid homing molecule CCR7. we next evaluated the antigen presentation of DC nanovesicles to naive CD8 positive T cells using OVA antigen. Syntheco-specific and H2KB restricted CD8 T cells were obtained from the spleens of OT1 mice and then co-cultured with DC nanovesicles. The DC vesicles promoted strong OT1 CD8 positive T cell proliferation and activation after 24 hours of incubation. In contrast, 293T nanovesicles and blank dendritic cell nanovesicles failed to induce OT1 CD8 T cell proliferation or activation. Upon successfully achieving antigen presentation, DC nanovesicles are expected to target lymph nodes and make full contact with T cells. To confirm the transport of DC nanovesicles into the draining lymph nodes, we labeled DC NVs with ICG dye and quantified the biodistribution after subcutaneous injection at the tail base. DC nanovesicles had efficient accumulation in the peripheral lymph nodes within 12 hours. The control 293T nanovesicles had a minimal fluorescence signal in the inguinal lymph nodes. On the other hand, C57 Black 6 mice inject subcutaneously at the tail base with a whole DC vaccine had a little signal in the inguinal draining lymph nodes. Due to the nanoscale size, DC nanovesicles apparently are more efficient to overcome tissue barriers and enrich in the lymph nodes than the whole dendritic cells. To address whether DC nanovesicles can elicit systemic T cell activation, we assessed the production of IL-2, interferon gamma, and TNF alpha by splenocytes harvested from mice immunized with nanovesicles or other treatment regimens. Obviously, the DC nanovesicle treatment demonstrates the highest concentration of relevant cytokines, the blue bars. Next, we investigated the induction of CD8 positive T cell responses by analyzing peripheral blood for Syntheco MHC1 tetramer positive CD8 positive T cells. The 293T nanovesicles induced about 6 to 7% antigen-specific cytotoxic T lymphocytes, CTLs, after the third immunization. Vaccination with antigen emulsified in aluminum hydroxide or alum had only 2% antigen-specific CTLs after priming. Yet no remarkable T cell expansion was observed after immunization for 3 times. In contrast, the DC nanovesicle group elicited a peak frequency of 26% of antigen-specific CD8 positive T cells after the third vaccination. The whole DC vaccine, DC vaccine had about 15% antigen-specific CD8 T cells. After 3 rounds of vaccination, the mice were then challenged with 200,000 HEP1 to 6 ova cells. Mice immunized with DC nanovesicles had no detectable tumor masses after more than 1 month. On the contrary, mice immunized with other formulas succumbed to tumors with only marginal survival benefit. We also observed different degrees of liver metastasis in the other groups at day 18 after HEP1 to 6 ova inoculation, whereas tumor metastasis was effectively prevented in the DC nanovesicle group. This study showed when we deplete CD8 T cells, it abrogated the immunotherapeutic effect of DC nanovesicles, whereas it's a red curve. While CD4 depletion, the purple curve, had much less impact on the DC nanovesicles. We also assessed whether the DC nanovesicles directly present antigen to CD8 positive T cells proactively, or delivers the antigen to CD8 positive T cells after being passively taken up by the antigen presenting cells, or APCs. We started this in the antigen presentation disabled mice whose B712 on APCs were knocked out. The DC nanovesicles still showed a strong resistance to tumors in the B712 knockout mice, while mice in the other groups were unable to inhibit tumor growth. Additionally, animals treated with DC nanovesicles had more peripheral blood CD8 positive T cells than the other groups. This data imply that DC nanovesicles can directly present antigen to CD8 positive T cells in vivo and activate a strong and effective antigen-specific CTL response without the need of APCs. This is very different from typical nanoparticles, such as liposomes. To prevent tumor immune escape by loss of a single mutant allele, we sought to elicit broad-spectrum T cell responses by employing multiple antigens, including M27, M30, and TRP2. Compared with a conventional multi-antigen alum or aluminum hydroxide vaccine, therapeutic vaccination with DC nanovesicles induced polyfunctional multi-antigen specific CD8 T cells and substantially delayed B16F10 tumor growth, but not complete tumor suppression. This could be due to the immunosuppressive tumor microenvironment featuring high expression levels of PD-1 on the draining lymph nodes, CD8 positive T cells, and its ligand PD-L1 on the tumor cells. We thus developed the so-called antigen self-presenting nanovaccine with immunosuppression reversal with the acronym ASPIRE. ASPIRE was engineered to break the immune-suppressive PD-1, PD-L1 pathway while activating the immune response. Anti-PD-1 antibody was expressed on the DC surface, and then PD-1 ASPIRE was prepared. Now, in the same B16F10 tumor model, the previous nanovesicles without PD-1 antibody on the surface showed delay of tumor growth, but not complete tumor regression. We also explored whether the co-delivery of B712 and the anti-PD-1 promotes immunosuppression reversal and reactivates the exhausted CD8 T cells to restart the cytotoxic T lymphocyte CTL response. We validated the result or effect using a Lewis lung carcinoma LLC model, which is sensitive to anti-PD-1 therapy. To eliminate the interference, we depleted CD4 T cells in the mice. We then assessed the production of granzyme B and the interferon gamma by tumor-infiltrating CD8 positive T cells. The CD8 T cells isolated from tumors in the PD-1 DC nanovesicle group had the highest percentage of relevant cytokine secreting CD8 positive T cells. The PD-1 DC nanovesicles elicited tumor regression in 8 out of 10 animals. In contrast, all mice receiving PD-1 DC nanovesicles with B712 knockout showed tumor progression instead. We further explored the impact of the ASPIRE integration strategy on the interaction between antigen self-presentation and the immunosuppression reversal. This was accomplished by using a murine MC38 colorectal cancer model. C57 black 6 mice were subcutaneously inoculated with about 100,000 MC38 cells and vaccinated with ASPIRE. The PD-1 positive, CD38 high, CD8 positive T cells have been described as a population of dysfunctional cells that fail to respond to antigenic stimulation and do not elicit effector functions. When ASPIRE was given, a significant decrease in the frequency of such cell population and a significant increase in the frequency of tumor-infiltrating antigen-specific CD8 T cells were observed. There was a significant improvement in the survival of mice treated with ASPIRE in comparison with those treated with other vaccine combinations. See here, this orange curve shows 100% mouse survival after at least 2 months. This ASPIRE nanovaccine platform showed the ability to activate the immune response and break immune tolerance. It is able to stimulate powerful CTL responses and enhance the immune checkpoint blockade with remarkable therapeutic efficacy. Based on the examples I showed here for infectious disease and cancer interventions, I hope I have convinced you that the biosynthetic cellular membrane vesicles can act as tolerable shells for delivering biofunctional moieties and therapeutic entities. I'd also like to acknowledge my previous laboratory of molecular imaging and nanomedicine, Minh, at the National Institutes of Health, and my current lab, Translational Nanomedicine and Theranostics, or TNT lab, at the NUS. My 2 frequent collaborators contributed a lot to this project. They are Gang Liu at the Xiamen University and Lang Rao at the Shenzhen Bay Laboratory. They were both my students before. And my funding support from NIH Intramural Research Program, NUS Startup Fund, Nanomedicine Translational Research Program of NUS School of Medicine, and NUS Center for Nanomedicine. Thank you for listening, and I would be happy to take questions.
Well, thank you very much, Shawn. Professor Chen, thank you for sharing that work. I think this is really an impressive amount of work that you've shared. It's also very useful against both infectious diseases and cancers. And so we have some questions that are starting to come in the chat box. And I would just like to emphasize to everybody, I know that we have an international group here. So if you don't feel comfortable sharing your question in English, feel free to use your native language. I think we have a lot of people who speak different languages here today, and we can somehow, I know that Minh has recruited a few folks who will also help to make translations if necessary.
And we have Google Translator.
Yeah, I think we have plenty of translators. So let's, I'm going to start with Patrick James. So Patrick, thank you for joining. I know that Patrick sometimes joins our, when we have it at a time that's good for the US, and he's joining in the middle of the night. So here he is. I'm glad to have you here, and more of a humane time for you. So Patrick, you have a couple questions here for Shawn.
Yeah, I can read the questions. He was asking about the dose of ASPIRE and the frequency of the application, and also about the PK from the second and third doses. Those are very critical questions. For the ASPIRE, typically, I think it's about 100 micrograms per injection. I might be wrong, but I can double check, but it's really around that range. Then how often they are applied? We probably did every other day. But again, what frequency is the best frequency? It all depends. Then the PK, no, we did not look very carefully about the second and third doses. We all know those immune response by different nanoparticles, the behavior can be very, very different. And that also happens to the vaccines, right? How come the second boost is much better? The third boost is enormous, right? So it's also possible for Aspire that if we do it properly, and we haven't done it, it could be even better. That's probably what you're asking, right? So if now it looks very good, but if we modulate properly, titrate the dose and the interval properly, and understand better the PK part, based on the PK, then decide what's the right frequency or the interval to administer. Then I would say, yeah, the second and third boosts would probably even be more exciting. In general, this strategy, I think there are one slide I want to highlight is that we do not require APCs. That's the key for this particular paper. That's how this paper will spend quite a few years, eventually convince the reviewers that, why not liposomes? Why not any other nanoparticles? Why not just this and that, right? They do not require APCs. That's the key. For typical nanoparticles, yes, you do. But for EVs, you may not need that either. We haven't tried DC EVs, right? So, but DC cell vesicles can be made relatively more easily, but still many steps of centrifugation, sonication, extruding through pores and so on. Yeah.
Thank you, Shawn.
Professor Lei, do you want to ask your questions too?
Hi, Shawn. That's great talk. And I'm so glad that you has come to NUS so that we and many of our colleagues had a chance to collaborate with you over here. I wonder if how well is the yield of the vesicles made from extrusion of cell membranes. Do you get a lot of them if you purify them from primary cells such as platelet? I recall that you purify some from the mouse platelet or macrophage. So how many mice do you need to get a large amount of these vesicles? And what's the topology of these vesicles? So you use some of the native protein on the membrane. Do you think that most of the vesicles have this protein on their surface, or some of them are inverted? And in terms of purification, how do you separate the fused membrane vesicles from the original vesicle and nanoparticle?
Those are all legitimate questions. Everything has some challenges, right? We talk about the bright side, and we often don't mention the dark side of the story, right? First thing is about the yield. The yield can be high because you simply get rid of the intracellular component. Then the cell membranes are disrupted. After disruption, we have done a lot of work trying all kinds of different surfactants. The surfactants help reassemble those fragments into vesicular structures. So that's the key, and we have spent quite a few years, and we are quite comfortable with all kinds of cells, forming those vesicles. You've seen several TM pictures. They do look pretty much homogeneous and consistent, right? Some are more like the DC vesicles, look very nice. very nice vascular, but some other cells, is somewhat thicker than some other more different cells. But yield is not a issue. Then the question about the protein orientation, right? Some proteins are sticking out, some proteins are inwards, right? When you try to form the vesicles, how do you make sure the desired proteins are all, pointing out? That's actually not difficult, because when you have those, in the very beginning, I only spent a few seconds talking about the integrin sequence to the signal peptide to make sure the proteins are properly oriented originally on the whole cell. Then when you form the vesicles, still the outer membrane remains the outer membrane. The inner membrane remains inner membrane. That's the way how they form the vesicles or miniaturized cells, right? So the proteins originally transfected properly, they will still orient the same way when they form the vesicles. So that's not an issue. Yeah. So you wouldn't have this disoriented proteins if they were properly constructed in the beginning. Did you have another question or something? Did I miss something else?
The question about purification, sorry. That's the
Sorry.
last question. The purification.
The purification. Yeah. So that's something tough, because you can. There's always cell debris, right? And so I think the challenge is to get nice vesicles and then get rid of all those chunks of cell debris. That's something always a challenge. I talked very often with Liangfang and others, because Liangfeng was like my little brother. They have the same situation. RBC is one of the best. Any other cell types, you will face certain issues. To be honest, you just cannot get rid of the cell debris 100%. There are always something there. So that may become a issue for
and not only cell debris,
scale
but
up
I wonder,
and
because you try to fuse several type of vesicle together. So how do you separate the?
How about the ratio, the different ratios, right? How do they become a homogeneous, right? Instead of being heterogeneous. So that's a challenge, right? So we even tried 3 or 4 different cell membranes, try to hybridize them together. We tried RBCs, or platelets, macrophages, tumor cells, or everything together, right? We haven't tried very hard to make them homogeneous. It can also be difficult to characterize how homogeneous, unless you have different fluorescent proteins or fluorescent tags on different cells. Then you try to hybridize and look at the overlap of different colors. So those are things we haven't dived into yet. If we are serious about the translation, then we need a significant amount of money to do this tedious work, and many students, to be honest, they can publish, but they don't want to spend all the time to study all the basics, because they may not get into publication easily, right? So I see Patrick nodding. But those are very critical questions for sure. Yeah, I totally agree. I think we can talk a lot more. We are also beginners, here, at least showing the proof of principle. I think all the 4 examples here, there are a lot more questions. These are simple, just, telling you we can do that. What the EVs can do, cell members can also do to some extent.
Yes, I agree.
All right.
Thank you so much for your answer. And I think you have
Thanks.
more questions from the audience.
Okay, I think, well, I may go to Yongliang then Patrick again, all right? So Yongliang asked the question, what happened to the nano decoys after neutralization, blah, 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 breakdown in phagocytes, a possible vesicle-mediated enhancement of viral infection? Great question again. We don't know what happened to the vesicles. Those experiments were extremely difficult to do last year during the pandemic because it took us a lot of effort to have access to BSL-3 lab. You know, it's a very political, very polarized environment, especially during the US-China tug of war, because we had the Long Rao in China tried to access BSL-3 lab. We need to get all the clearance from federal government because I'm a federal employee, and the Chinese government did not want any of us to enter any of their facilities, right? That doesn't sound unfamiliar to any of you at all, right? So, you know, we only had a few very, you know, superficial experiments because the reviewers keep bugging us, says, you know, how can you not have access to it? Because it feels like you're an almighty person. You can have all the connections with the Chinese government. You should be able to do everything you can. No, the answer is no. Because of the connection, the Chinese government doesn't want us to do anything, because anything we do can be against to whatever is ongoing. Gosh, this goes on and on. It's just ridiculously difficult. So we had only 1 or 2 experiments at the cellular level to deal with the virus. And for animal models, we only had the acute lung injury model. So we really did not have time to answer that question, but it's a great one. Not sure if we apply those nano decoys in vivo, most likely eventually will be engulfed by macrophages. I'm sure most of those will be, you know, phagocytosed. Right, I'm not sure I did answer his question. So there are many more questions come up. I open the can of worms now, right?
Yep, thank you.
So John Wei has a question about how often the mice will be vaccinated and how long it lasts. Usually vaccination in mice, we do weekly vaccination for 3 times. 3 times is long enough, then the effect can last for many months, actually. The longest we have seen in a tumor situation, it's up to, I think in mice, we have tested up to 200 to 300 days. So it's really, it can be long sustaining, you know. Also, it comes to whether it's prophylactic, you know, preventive vaccine, or it's a therapeutic vaccine. For prophylactic vaccine, the effect is much longer. For therapeutic vaccine, the effect will be somewhat shorter. All right, sorry, I need to speed up because Georgia has the question about how do we normalize nano decoys to suppress pneumonia? Right, great question. Right, so how we normalize? We actually look at how much we can neutralize. We use the mass amount of the nano decoys. We use some kind of you know, rough measurement based on UV to know the amount of nano decoys. Then we do have data to show, you know, for example, 100 micrograms of nano decoys can neutralize how much interleukin-6 or GM-CSF. Those actually we have number in the PNAS paper. Right, so micrograms of protein, and then nanograms or picograms of interleukins, right? Yes. Then
for the memory coding.
Right, toxicity and a deterioration to cardiovascular diseases, which needs to suppress inflammatory response. Yes, so it's always the again the you know, contradictory to each other. You know, for often for cancer, you want to stimulate the inflammatory response. For cardiac, you want to suppress
确
the
实，心脏
inflammatory.
和癌症就是一个矛盾体。
Yes, indeed. Now, that's Chinese, but she's saying this is a a contradictory existence. Whatever, right? So it is tough, right? So I think it goes too deep for this talk. It's about for example, cancer patients when they have the cardiovascular complications. Then how do you use immunotherapy to treat those patients? I think it's not only relevant to EVs or nanovesicles at all. It's more of immunotherapy in general, about the cancer versus cardiovascular complications. Ramesh, right? How to make sure the membrane coating integrity in vivo? Great questions. We don't know. We hope so. And just like the same question, how do you confirm the liposomes are intact in vivo? They may or may not, right? So all depending on how long it takes to disintegrate. I'm sure all those EVs and membrane vesicles like liposomes will fall apart over time. And that caused the question of, often I'm an imaging person by training, what you label and how you label and how you study the PK, the biodistribution. There's discrepancy between the label distribution versus actual particle distribution, which we really do not know most of the time. We're just assuming what we see is what we believe. Right, so there's again, excellent question. Is probably another PhD thesis project or multiple PhD thesis project. Sorry. So this person, the third signal cytokine, also play an important role to shape the naive T cell during the antigen presentation process. How they aspire to control the native T or naive T cell native T cell differentiation. Which third signal cytokine are we talking about? I'm not sure. Maybe I can talk to Bolo offline. He's from John Wei's lab. Yeah. Sorry. Did I miss other questions? No, too many questions. But anyway, if I feel sorry if I did not answer your question. And again, I have to confess I'm a chemist by training. I'm not an immunologist. Don't torture me with all the immunology questions, and I'll be happy to answer offline or find the people who did the work to address the questions. So how stable are the nano decoys? Can they be stored at the 4 Celsius degree or have to be stored at -80? No, not -80, because once they are disrupted, they're not live. They're cell debris, and the cell debris formed the cell vesicles, so they are stable. So the way we store liposomes is just fine for the EVs or nano decoys whatsoever. 4 degrees is perfect. It can be stable for many months, right? As long as it reaches the CMC critical micelle concentration again, because just to think it's like it's very much like lipid nanoparticles. There's no magic. The only difference is such cell membrane vesicles are more complex than liposomes. They could have more functions than liposomes, but you treat those as liposomes, then it'll be just fine. It could be lyophilized. Once lyophilized, then when you reconstitute, it can be a challenge depending on how. Usually for liposome formula, use a Onivite as example. That's a liposomal formula for pancreatic cancer treatment. It is a emulsion. Usually they are not lyophilized. They are kept as an emulsion.
So, Shawn, I think that we're coming.
You're right. We are over time.
But I would just like to ask a question here that comes from, I think, some of the hardcore people in the EV field. because I think that we can separate some of the EV researchers into the splitters and the lumpers. So the splitters say we have all these different little, little categories of EVs, and the lumpers say, if it's an EV, it's an EV. It doesn't matter where it came from. So do you think that there would be a functional difference in a nanoparticle that you have coated with, let's say, an internal membrane from the cell versus a surface membrane from the cell? So the difference between the so-called exosomes and ectosomes or microvesicles, do you think that would do anything?
Finally, this question came. I was expecting this question all the time. That's the most fundamental question, right? Yeah, I'm also wondering how much different. It goes philosophical pretty much, because we don't have enough data to support. We have to be religiously believing whatever we are talking about here. So EVs, if we believe they will originate from cell surface or plasma membrane, they're supposed to maintain most of the proteins from the plasma membranes, probably no more than what's from this plasma membrane, right? And the cell surface, the membrane vesicles, we artificially, mechanically, to form EV-like structure. If we say it this way, probably it's half right? Because you are just probably accelerating the formation of quote unquote EVs through disruption, surfactant, messing around to make those. They probably do not have too much difference in terms of the protein content or even the protein distribution pattern. That's my gut feeling. It's just the EVs are formed, by smart cells intracellularly and then spit them out, right? So they probably look better and more uniform when we do it, using external force to form cellular vesicles. They would always be less homogeneous. Some disruption may have the shear stress on those, whether those stress or the surfactants have additional effects, the bad effects on those, unclear. So I would be a little bit cautious. I kind of feel that the EVs and the cell memory vesicles can be quite similar, not vastly different, but we can make more if we can afford losing some of the integrity, homogeneity of the structures. We can make large quantity, right? But of course, which parts of the cell membrane become the EVs, that's still a myth, right? So I don't know. It's just my guess here.
Yeah, I think there's a lot more that we need to understand, and I think
Correct.
that even the nanoparticle approach, beyond, in addition, I guess, to what you can do with the therapeutics there, it might help us to answer some of the questions about the basic biology as well. So,
That's true. I purposely did not even want to talk about the basics, and also for the in vitro diagnostics, a lot of applications, but as delivery vehicle, as the using those like nanoparticles for intervention. From this aspect, EVs and cellular vesicles have a lot in common.
Indeed, and that's what I'm taking away from this this session today. That I think the communities of EV research, nanoparticle research are not distinct anymore. We are making ties with each other. We're discovering things together, and that will take us into, I think, a very bright future of these nanotechnologies. So Shawn, Professor Chen, thank you so much for joining us today and for sharing your knowledge. Professor Minh, thank you for co-hosting with me today. We're going to look forward to our next session at the beginning of July. So stay tuned for emails and advertisements about that. Minh, did you want to say anything more about the next session here?
Yes, we welcome you back to attend the next session. On the 10th of July, we'll have Xiao Ning, who will talk about extracellular vesicle for diagnosis. And we're very excited to hear from her about the latest development in her microfluidic chip that's very sensitive for diagnosis of cancer and Alzheimer's disease.
Excellent. So
thank you so much, Ken, and thank you, Shawn.
Another good session planned then. So thank you all for joining today, and I wish you all a very productive Friday and a good night for those of you who are more in my time zone. And thank you all, and look forward to seeing you again soon. Take care, everybody. Bye now.